Can I start again?
Yes, sir.
Yes, sir.
Okay, today we will study about the replication in the eukaryotes. So eukaryotes origin has two components. One is a core component and second is an auxiliary component. As we have discussed in the prokaryotes, in the bacteria and phage virus, the replication will always start from the specific site of the DNA that is called origin. So the same way in eukaryotes, replication will also start from origin. So this origin has two components. So one is a core element that is made up of three regions. That is a origin recognition element. Second is a DNA unbinding element, and that is a AT rich region. So that is a full form is written here: origin recognition element or DNA unbinding element. So if we combine these region, that is a core component and that is a auxiliary component. So auxiliary component has also divided into two parts, auxiliary 1 and auxiliary 2, or this is core component. As in the previous slide we have seen, core component is made up of origin recognition element, DNA unbinding element and AT rich region. So this is core component and this is both are auxiliary component. So these all components form the origin of the DNA in the eukaryotes. So this is one origin. If we summarize this, this is total one origin which is present in the DNA. So this is the DNA and this is whole is a origin. Because we are studying about the multicellular organism like human or eukaryotes. So in the multicellular eukaryotes, the DNA is very complex and very large DNA is present. So because due to the presence of large DNA, the multiple origin are present in the eukaryotic DNA. So that means the replication will start from the different sides of the DNA. It will not start only the one side because multiple origin are present in the DNA. Suppose this is a one origin, and if you go in the DNA, so after some base pair, another origin will be present. Then if we go further, then another origin will be present. That means there are a lot of origin are present on the DNA. So that is a special quality of the eukaryotic DNA. So replication start in the eukaryotes when origin recognition protein will bind with the origin. So origin recognition protein will bind, especially origin in a specific region of the origin, that is called core region, and ORE, that is origin recognition element. This is the main component of the origin. And now what is the function of auxiliary element? This auxiliary element will bind with the transcription factor, and this transcription factor will also bind with the origin recognition protein. So basically transcription factor is take part in the transcription. Transcription is another process that take part in the formation of the DNA to the RNA. So we have just described that in the transcription process, the transcription factor are required, and these transcription factor also bind with the origin part, and which part of the origin, especially auxiliary component of the origin. So in the replication, we are not discussing about this transcription factor. In replication, we are discussing here the origin recognition protein that will bind with the origin. So you have to keep in mind this structure, this core region and auxiliary region. And now we can discuss some points about this. Like auxiliary component provide the site of transcription, and core component is analogous to the transcription promoter. Actually, what is the transcription promoter? We have not studied yet about the transcription, but if you study in the next unit about the transcription, so as the replication start from the origin, and in the same way the transcription start from the promoter region. So that means promoter is also a specific sequence which is present on the DNA where the transcription will start. So we are telling that here in the eukaryotes core component is a part of the origin. So core component is analogous to the transcription promoter. That means that has same function as the transcription promoter. So what is the function of promoter? To start transcription, and what is the function of core promoter? To start the replication. So that is analogous. Analogous means same function, that is a starting function. So it will start because replication, and it will start from the transcription. So another component of this, I have already explained about the transcription factor have two domain. One is a DNA binding domain and another activation domain. So here that is a transcription factor that has two domain. One is a DNA binding domain. So with the help of DNA binding domain, it will bind with this DNA portion. And second is a activation domain. Activation domain involve activation of origin recognition protein. So that is a origin recognition protein. So it will activate the origin recognition protein. Two domain, one is a DNA binding domain, so bind with this auxiliary component, and second is a activation domain, so it will interact with this recognition protein and activate of this protein. So because this is transcription factor is a protein also, and this is also protein, so this activation is done by the protein interaction. So that means this transcription factor interact with this protein and activate this protein. So this process is called protein interaction. So in the replication, only the function of this transcription factor is just activate the origin recognition protein. So when it will activate this protein, this will bind with this ORE or origin recognition element, and the replication will be started. This is the role of this transcription factor here. And now another point, in the eukaryotes, origin recognition element, that is a ORE, have ARS sequence. AR is a autonomic replicating sequence. So portion ORE is the main portion of this origin, and it contains the autonomic replicating sequence. That is very common sequence, ARS, autonomic replicating sequence. So this present in the eukaryotes, and this is also present in the yeast. In the yeast, it is very common autonomic replicating sequence, and also there are many types of the autonomic replicating sequence. So we can see that autonomic replicating sequence is like the origin, because as ORE is like an origin, in the same way autonomic replicating sequence is also an origin. So this is the ARS, autonomic replicating sequence, and it is responsible for the origin. So in some cases where this whole structure is not present, like DNA unbinding element, origin recognition element, this auxiliary component, if these all structure not present, so only the structure is present ORE, and in the ORE there is a sequence that is an autonomic replicating sequence. So in some species, some animals, only ARS is present. So ARS is responsible for it is like the origin also. So we can say that ARS is a autonomic replicating sequence or origin. So replication will start from the ARS. Now some important point about the replication, that is what is the replicon. So simply the unit of the DNA in which indivisible act of replication occurs called replicon. That means the portion which is responsible for the replication of origin replication, that is called replicon, or we can say that the origin portion of the DNA, that is also called replicon, because the DNA is starting from the origin. So the unit of the DNA in which indivisible act of replication means simply replication occurs. The unit of the DNA where the replication will start, that is called replicon. So in the replicon is usually applied in the eukaryotic chromosome. We have not studied about the replicon in the prokaryotes. So we are talking about this word. We are using this word especially in the eukaryotic chromosome or origin. Eukaryotic replicon is relatively small. The rate of replication is much lower. In that, the rate of bacterial replication fork moment. So here, that is another point, the rate of replication, because replication starts from the replicon or origin. So the rate of replication is slower in the eukaryotes in compared to the bacteria, because in the bacteria replication fork moment occur, but in this cases replication fork is not showing, but the rate of replication is slower. Although The mechanism of replication in the eukaryotes is not described fully. That means there is very few knowledge of the eukaryotic replication. So we are not studying the step-wise step that this protein will come and bind here and it will start. So that is why we are just study about the general feature, whatever the known about the replication. Like what is the known about the eukaryotic replication? It is known that like the origin, uh, this eukaryotic chromosome origin have these structure, core element and auxiliary element, and core element has origin recognition element. So this element have autonomic replicating sequence, and this origin is also called replicon. And now this is the structure of ARS, autonomous replicating sequence. So as I have described about this autonomic replicating sequence is present in eukaryotes or in the ORE region, origin recognition element or origin recognition complex. So whole structure is present on the origin recognition complex. So ORC is just like a ORE. I have written here ORE, so you can also write here origin recognition complex. So this is the main portion because autonomic replicating sequence is present in this region. Same thing, either you can origin recognition element or origin recognition complex. So origin recognition complex. So in this portion, if we talk, this ARS has this ORC region, and ORC contain B2, B1 and ACS. That means ACS region and B1 region and B2 region. These come into the origin recognition complex, and there is also domain that is a C1 and B3 domain. So this is just for learning that ARS, how many domains are present in the origin autonomic replicating sequence, and in the autonomic sequence, what is the ORC or origin recognition complex. So this portion is important. So this portion is called autonomic replicating sequence. So why it is important? Because all the protein will bind with this region B1, B2 and ACS. So the initiating protein in the replication, that is a MCM protein, mini microchromosome maintenance protein. So origin recognition complex is act as a loading pad for many protein like MCM. That means this portion will be responsible for binding of all the initiating factor or all the protein which start the replication. So which protein? Like here I have shown one example, that MCM protein. So when the MCM protein bind with this region ORC, then replication will be started. So what is the MCM? MCM is a mini microchromosome maintenance protein. So we have to learn these two structure, one ARS, autonomic replicating sequence, and second this origin portion. Okay. Eukaryotic initiator. So now we discuss about what is the eukaryotic initiator. So eukaryotic initiator. Yes.
सर वो B3, B2, B1 क्या होता सर? ACS?
ये जस्ट एक डोमेन्स हैं जो उसमें प्रेजेंट है ARS रीजन में. अभी मैं यहाँ पे इसका फंक्शन बताऊंगा आपको कैसे, मतलब क्या काम होता है, और इसके बारे में बहुत ज़्यादा नोन नहीं है. दैट इज जस्ट नेम दे रखे हैं. These domain are present in the ORC. Actually, what happened in the yeast? In the yeast, this is autonomic replicating sequence. So some factor will come and bind with the B2, and while in some another organ, some factor which bind with the B1. That is this domain. This is separate domain, and these are present in the ORC region. So what is the difference in these domain? In some eukaryotes, ACS is a main domain. In some is a B1 or B2, or maybe possible that it is for the regulation of the replication, because there is not too much knowledge is present about these domains. So we are not discussing here the function. So we are just displayed here. This is the structure of the ARS, and if it will require, then I will explain some important points. But this is not relevant here. So you have to just learn about this. origin recognition pad is these three domain B1, B2, B3 and ACS. So this portion is a key ORC region. So that means all the initiative protein that will come, that will bind of this particular region. We will discuss here. Like first we are talking about the initiator. So initiator protein in the eukaryotes is a MCM protein. Like in the bacteria, that will initiation protein DNA A protein, or in the phage initiation protein is a RAP A protein or something different, DNA J protein like this. But in the eukaryotes, the initiation protein is MCM. MCM is a mini microchromosome maintenance protein, and initiation factor and different type of the initiation factor that are not known. So MCM is a mini microchromosome or plasmid maintenance protein, and the MCM. So what is the function of the MCM here? I am just describe here. The MCM protein are essential for the maintenance of the ARS. So that means for the maintenance of this autonomic replicating sequence, the MCM protein is required. So in its origin site, MCM protein phosphorylated. That means when the phosphorylation occur of this MCM protein, then replication will be started. So how the phosphorylation occur? Phosphorylation means addition of the phosphate. If I am talking that MCM protein will be phosphorylated, that means the phosphate group will added into the MCM protein. So that means its phosphorylation occur. So phosphorylation occur with the help of some enzymes. Different type of enzyme is required that will cause the phosphorylation, or that will add the phosphate on the MCM. So which type of enzyme is required? I have given here example, like mainly the main function of the kinase enzyme. So kinase enzyme always add the phosphate group in the any protein. So the main function of the kinase is a addition of the phosphate, or in the scientific term we can say that function of the kinase is phosphorylation. So kinase enzyme or another some chemical compounds, or that is called cyclin, and there are many type of the cyclin, like A cyclin, B cyclin, C cyclin. So cyclin B CDK, cyclin dependent protein kinase. So you can just learn this cyclin B CDK that will bind and initiate the replication. So that means in the initiator protein, some MCM protein will come and kinase enzyme come, cyclin BCDK come, and these proteins will come, and it will cause the phosphorylation of the MCM protein. And when the phosphorylation of the MCM protein occur, then initiation will be started. So till that time only this is the known fact how replication will be start. It is known that only some replication protein present, especially MCM protein, and when MCM protein will be phosphorylated by the kinase enzyme, then this process will be started. So for the phosphorylation, not only the kinase is required, some cyclin dependent protein kinase is also required. So some cyclin dependent kinase and kinase enzyme that will come and cause of phosphorylation of MCM, and the replication will be started. तो यहाँ तक क्लियर है आप लोग को? एक बार फिर से रिवाइज करा देता हूँ मैं पूरा प्रोसेस को. Origin of the replication start from the specific type of the sequence that is called origin. So in the eukaryote, origin is made up of two components, core component and auxiliary component. Core component has these three regions: origin recognition element, DNA unbinding element, and AT region. Core region, and this two is the auxiliary component. So this is made the origin of the in eukaryotes. So in the process of starting this transcription factor, that will has two domain. One is a DNA binding domain, so it will bind the DNA especially to the auxiliary component. Another is the activation domain, so it will bind with the origin recognition protein. So what is the origin recognition protein here? The origin recognition protein is a MCM protein. So we are talking about this origin recognition protein, MCM protein or cyclin protein, cyclin CDK. These are all are called origin recognition protein. So how this will activate? When transcription factor will activate this MCM protein, or these different protein will come and phosphorylated of the MCM protein, that it will bind with the origin recognition element and it will start the replication. So Replicon is just definition. So another part is the autonomic replicating sequence. So autonomic replicating sequence is a specific sequence where the replication will start, and this sequence is present especially in the central portion of the region that is called origin recognition element, and it will start the replication. So I have already explained about the MCM. Now come to the eukaryotic DNA polymerase enzyme. As we have discussed that in the prokaryotes there is different type of the enzyme, DNA polymerase 1, DNA polymerase 2, DNA polymerase 3. In the same way, the eukaryotic DNA polymerase had DNA polymerase alpha, DNA polymerase beta, and DNA polymerase gamma and delta. Okay, so then in the next class we will discuss about this all the replicating enzymes in detail. So I am just ending here.